So what Okazaki was able to figure out is that instead of one long continuous new strand, like we see with the leading strands, the lagging strand will be a series of short little fragments that will be joined together to form one bigger and bigger strand later. These fragments, as we see here, will be made in the correct orientation away from the replication fork, oriented back towards the origin of replication, and that at a later point an enzyme will come through and start joining these fragments together, making the new longer and longer daughter strand. What we now know happens on the lagging strand, somewhere away from, near the replication fork, oriented pointing towards the origin, primase will come in and lay down a primer, okay, because DNA polymerase, DNA Pol 3, has to have that 3' end right here, something to build on. DNA polymerase will come and start laying down a new strand of DNA. Read the template, make the complement. And notice it is doing it in such a way that it is going towards the origin. And I'm trying to write origin, and it's really crappy. So the origin is somewhere over here. And it is making the new strand in that direction. Remember, the leading is going from the origin towards the replication fork. So now we're replication fork. Fork is over here somewhere, and it's moving away from the origin. So everything is backwards here on the lagging. Once it hits a preceding fragment or it hits the leading, it makes a small, short fragment, an Okazaki fragment. DNA Pol 3 jumps off, goes backwards. Think of it as leapfrog, reverse leapfrog. Goes until it hits a new primer and then fills in. Here's a second fragment. It'll jump off, move back to start making a third fragment, and so on and so forth. These primers you see right here are a problem. DNA Pol 1 comes in, fills in, clears out, ligase, joins everything together. This is why it's called a lagging strand. Look at all these multiple steps that have to occur for every one of those thousands upon thousands of fragments that are going to have to be made and then ligated together to form the new complementary strand on the lagging, for the lagging side of the replication fork.